Zane, the podium is yours.
Thank you, Alice. Hi again, I'm Zane Hauk, and I'm a research scientist at ZRT Laboratory. I'm going to briefly talk to you today about method validation, specifically for methylmalonic acid, or MMA, which is a biomarker for vitamin B12 deficiency. Vitamin B12, or cobalamin, is a fat-soluble vitamin not produced by the body, thus it's only gotten through the diet from dairy, meat, and other fortified foods. B12 is important for neurological and cognitive function and is a factor for two enzymes. The first is cytosolic methionine synthase, which is used in the methylation pathway to convert homocysteine to methionine. The second is mitochondrial methylmalonyl-CoA mutase, which is part of the citric acid or Krebs cycle, and is a converter of methylmalonyl-CoA to succinyl-CoA. Here I have developed and validated a method for LC tandem mass spectrometry analysis of methylmalonic acid in dried urine following FDA guidelines. Dried urine is a great clinical collection tool because it is non-invasive and it can be performed at home and mailed into a lab. MMA is a great indicator of B12 status among many other analytes because it builds up when B12 levels are low. Briefly, our method uses 3 6-millimeter punches, which is 37.5 microliters of urine, which is then extracted from the filter paper, filtered, a creatinine measurement is obtained, and then the extract is then shot into HPLC. Here I want to go through the steps of validating a laboratory developed test and go over our results. The first parameter we look at is precision. Precision is a measure of how consistently you get the same measurement. Intra-assay precision measures the same sample injected many times over the same run, while inter-assay precision measures the same sample over multiple runs. The FDA recommends that you have precision CVs of less than 15%, which you can see in the top right that we accomplished. The next parameter is accuracy. Accuracy is a measure of how close you get to the real value. For this, we have to send our samples to other reference labs to compare our results. It is not pictured here, but we did compare favorably to 2 other labs that perform this assay. Because dried samples is kind of a niche, we also had to correlate liquid urine to dried urine samples. And for this assay, we showed great correlation between the 2 different types. Then you have linearity and recovery. This verifies that you have a linear response over the analytical range and consistent recoveries over the full concentration range, preferably greater than 85% recovery. As you can see from our graph, we had great linearity and we had recovery of greater than 98% across our analyte range. Next up is stability, and this is usually the longest part of validation and requires at least 30 days and can take up to 2 or 3 months depending on how long you want to store your samples. Short-term stability ensures that the extracted sample is stable, whereas long-term stability ensures that the samples are stable under normal shipping or storage conditions. As you can see in the bottom right, our samples are stable up to 32 days on dried filter cards and up to 24 hours after they're extracted. Finally, you have sensitivity and selectivity. This determines your lower levels of detection and quantitation, as well as identifying possible interferences. For this assay, sensitivity is a little less important than it usually is because with B12 deficiency, you have elevated MMA values. With that being said, we still managed to get an LOQ of 0.1 micrograms per milliliter, which is lower than any other labs that test this. Selectivity for this assay is very important because succinic acid is an isomer of methylmalonic acid and is also an endogenous compound. So I had to ensure that I got baseline chromatographic separation of the 2 analytes, which I was able to achieve in less than 1.5 minutes of runtime. When doing clinical work, it's also important to establish reference ranges for your analytes to know what is a healthy population and what is not. We report our values in micrograms per milligrams creatinine with a healthy range of 0.03 to 2.6 micrograms milligrams creatinine. So that's it. These are the steps needed to validate a laboratory developed test. Thank you very much for your attention.